Hello and welcome back to A Level Biology Help. So this is another required practical video. So this is required practical 7, which is use of chromatography to investigate the pigments isolated from leaves of different plants for AQA A Level Biology. So before I start, it's probably a good idea that you're sort of familiar with the photosynthesis topic. So I will put a link to that video in the corner. Right, so first off, so just a little bit of background to the practical. So obviously the purpose of it is to use chromatography, or well, more specifically paper chromatography, to investigate and identify pigments in different plant leaves, because as you'll know, there are loads of different types of pigments that exist in a single plant. Obviously, because obviously so that they can capture light at different wavelengths. So yeah, you use chromatography to identify these pigments. And in chromatography, well, I will take you through the method in a bit, but the photosynthetic pigments are separated, and then you use something called an RF value to identify the pigments, which I will explain later on in the video. Right, so the method to this practical then, so it's a pretty simple one, pretty boring to be honest. So obviously you have to collect your leaf sample first. It's a good idea to use a plant that visibly has multiple pigments. But yeah, you obviously have to collect your leaf sample, and then you have to grind it up, obviously using a pestle and mortar and something that will break open the cells, such as acetone, and that will release the pigment so that you can investigate which pigments are present. And then you get a piece of chromatography paper, which it's important here that you draw a line about 1 centimetre from the bottom, and it's very important that you use a pencil line, which I'll explain just in a sec. And then once you've drawn your pencil line, you use something called a capillary tube, which is a really, really small, thin glass tube, very easy to break. I've broken quite a few in my time. But yeah, use this capillary tube to transfer your pigment onto the pencil line in the middle. So it will, this is just a schematic representation because, I'm doing this from home. I don't have access to a lab. So you have your piece of paper there with your pencil line and then your dot of pigment right in the middle. So yeah, as I said, so it's really important that you use pencil to draw your line. So this is something that is sometimes asked in exams, why is the line drawn in pencil? And this is because, well, if you used ink, that would obviously smudge, because it would leak. And, that would obscure your results. Obviously, if it's smudged, because you might misinterpret the ink smudge as a pigment. So yeah, after that, so what you do is you put your paper in, say, something like a measuring cylinder or a beaker in solvent. Well, these solvents can be, multiple different things. But yeah, it's important that you make sure the solvent doesn't touch the pencil line because, well, obviously if it touched the pencil line, then the pencil line could smudge as well. And what you do then is you just leave it until the solvent has run near to the top of the paper. Make sure it doesn't run, right to the top because, well, I'll explain in a bit. So when it's reached near the top, you have to obviously remove the paper and then draw a line where the solvent has moved up to. And this is kind of a representation of what it might look like. Obviously, as I said before, it's a schematic because I'm not in a lab. So you have your pencil line and then, well, the grey bit kind of represents the solvent and, you can see it's moved near to the top. And you'll see something that kind of looks a bit like that. So you'll have kind of different smudges of, different pigments. It probably, it won't be as clear as that, but yeah, it's just a schematic. So yeah, different pigments will separate and will appear as different spots on the paper. And how you identify these pigments I will explain in a bit. So your results then. So you have to calculate something called an RF value. So how you calculate an RF value is you divide the distance travelled by the solute by the distance travelled by the solvent. So the solute is your pigment spot, and the solvent is the liquid that you put the paper in. And obviously, the distance that it travelled by the solute, so your pigment spot, you have to measure from the centre of the spot. Well, you have to try and measure from the centre of the spot anyway. And how you identify your pigment is by comparing the RF value from your experiment to a known value in a database. So, for example, I don't know if chlorophyll A, which is a type of pigment, has an RF value of, I mean, this isn't the case, but say if it's like 2 or something, and you have from your experiment a spot of pigment that is kind of around that value, you can kind of assume that that pigment is chlorophyll A. But this is really important. It's really important to make sure that the RF value database that you use, because there are different ones, uses paper chromatography, so uses the same equipment as what you've done in your experiment, and make sure that it also, your database has used the same solvent that you've used as well. So obviously I've said that different pigments will have different RF values. So what causes different RF values? So obviously you have your affinity to the chromatography paper. So affinity is basically, it's another word for attraction. So the pigments that have a lower affinity will obviously travel further up the paper because, it will kind of repel the paper. So obviously it will move further. And also solubility. So pigments with a higher solubility will travel faster and therefore further. So this is something that I thought I'd add in because it's something that you can get asked in exams quite a lot. So especially in your paper 3, because your paper 3 has a lot of questions about, practicals and kind of general kind of knowledge around experiments. So I thought I'd explain or list for you some of the advantages and disadvantages of using paper chromatography to investigate photosynthetic pigments. So if you think about it, so the advantages, so obviously the apparatus or the equipment that you use is, it's simple stuff, and it's cheap, and it's quite a visual experiment, because you can see the different pigment spots, so that the pigments are visually separated, and it's useful for identification. And then the disadvantages then, so it's not very precise because, I've put there, it's affected by environmental conditions. So, if there's a higher temperature, for example, the pigments might move at a different speed, and obviously, that will affect your results as well. It's qualitative as well, so I mean, obviously you can't really properly estimate the all the pigments that are in the plant, just kind of some of them really, and you can't really, it's not totally accurate. Also, some of the pigment spots can overlap as well, especially if they have similar RF values, or they absorb light at similar wavelengths. And also limited resolution as well, because I mean it's not completely clear. And also your solvent choice affects your results as well. So depending on which solvent you use, it might give different RF values, and that can cause discrepancies in your data. So obviously identifying pigments, that's useful because, if you can figure out the RF values and then identify your pigments, you can kind of then figure out what wavelengths of light your plant absorbs at, because obviously, different pigments, they work by absorbing different wavelengths of light. So you can then use that to optimize conditions for plant growth, which obviously has, advantages in loads of different things. Right, so just a quick risk assessment. It's pretty much common sense, but I thought I'd take you through it anyway. Um, so yeah, obviously, your first hazard there is your solvent that you use, both in the chromatography part and also, um, grinding up your sample as well, because they're very, very flammable. And also, it can cause skin irritation and also inhalation hazards as well. So obviously, to control that, I mean, it's important that you keep away from naked flames. Keep solvent in a fume cupboard whilst you're not using it, and also, opening windows, just generally ventilating the room that you're doing your experiment in. And also, the general stuff like avoiding contact with skin and, wearing goggles and gloves, things like that, really. Um, Another thing as well is obviously there's a biohazard because, you're using a plant, and also the soil that the plant's coming from, because, things like that, they can have, harmful bacteria in them. And also, there's a possibility that you can be allergic to the plant or the soil that can cause, skin irritation, things like that. So obviously the main precaution to take there is to, wash your hands before and after doing the experiment. Okay then, so lastly, I want to just take you through a couple of exam style questions that are sort of related to this practical. So we're just going to go through this one first. So I'll read it out. So a student used chromatography to separate the different photosynthetic pigments in a chlorophyll solution. She had the following materials. So your chromatography paper, a ruler and a pencil, suitable glassware, so that's what you put your paper in, a solvent, so they've called it solvent A here, and 2 centimeters cubed of the chlorophyll solution. So describe how she could use these materials to separate the photosynthetic pigments by chromatography. So these are pretty common questions you can get, just kind of doing a brief outline of the experiment. So it's worth 4 marks here. So you're going to have to pretty much write it in kind of 4 steps, really. So this is the answer I've provided. If you want to have a go at it before, then feel free to pause the video. But this is what I've said. So you have to draw the line. So it's called the line of origin. It's that's kind of one of the terms that is helpful for you to uh, remember. So the line that you draw on the paper is called the line of origin. So I've draw the line of origin with a pencil on the chromatography paper. And then you add the chlorophyll solution to the line, and then you add the solvent, making sure that it remains below the line of origin. And then you remove the paper before the solvent reaches the end of the chromatography paper. So that's kind of an outline of the experiment. So I've kind of done 4 points. So you draw the line of origin on the paper, you add the solution, you add the solvent, and then obviously I've added an extra point about making sure that the solvent remains below the line, and then obviously remove the paper before it reaches the end of the chromatography paper, because that kind of marks the end of the experiment. So if we look at the mark scheme then, so obviously the first marking point, so yeah, draw the line of origin on the paper. So you don't actually have to state that you have to use a ruler and a pencil, because it's a describe question, so you don't have to explain why you do something. So that kind of extra detail isn't that important, but I think to be sure, that you're getting the mark, it's ideal to remember to write, you draw it with a pencil. So the things that are in brackets, you don't have to put to get the marks. And then you add the chlorophyll solution to the line. Obviously, you don't have to mention, use a capillary tube or a pipette or whatever to add the solution. And I didn't say that. You don't have to say that to get a mark. So yeah, I'd get that mark. And then you add the solvent or solvent A below the line because, I've stated that that was really important when I was talking about the method. And then it says, so except so solvent is not above the line or solvent below line. So, well, obviously that's kind of similar to what the marking point has said, but yeah, that's what they accept for that mark as well. And then remove before the solvent reaches the end of the chromatography paper because that kind of marks the end of the experiment. So that's something important to write. Or you can write mark where the solvent reaches the front. So obviously the front basically means that position where it's reached near the end of the paper. And then the other things that it accepts as well. So you can actually draw the experiment and obviously label it. I'm the type of person that just prefers to write. But if you're the type of person that likes diagrams, and feel free, it does kind of take a lot of time, but yeah, feel free to do that. They do allow filter paper instead of chromatography paper. It's not really filter paper, but yeah, they do kind of allow that, so you would still get the mark for that. And they accept remove when the solvent reaches the end or the top. Well, immediately, because, you wouldn't want it to kind of go over that, because obviously that would obscure your results. And then they also accept for that fourth marking point, except ensure the solvent does not reach the top or the end. That's kind of not really a point in the experiment yet. It's an important point that you don't really want the solvent to reach the end, so they do accept that. And then ignore the RF values because so you wouldn't get a mark for saying that, because that's not really what the question's asking you. The question's asking you to describe how you would separate the pigments. It's not asking you to describe how you would distinguish between the pigments, which is what you would use an RF value for. Alright, so the last one then. So using solvent A, the students separated 5 pigments, so they found 5 different pigments came about with this experiment. And then they repeated the method using a different solvent, so solvent B. So using solvent B, they separated 6 pigments. So as I said before, different solvents can produce different results. So solvent A, they got 5 different pigments, and then solvent B, they got 6 different pigments. To explain the difference between the results. So, as I said earlier on in the video, there are lots of different things that can affect the separation of the pigments. So, I was talking about solubility and affinity. This is what I've written. So, one pigment was soluble in solvent B, but not in solvent A. So, obviously, if something's soluble in a solvent, then obviously that would cause it to kind of separate and, on the paper and kind of go up with the solvent. So that would, cause a different pigment to be separated. So yeah, so the more soluble pigments that are soluble in that particular solvent, the more pigments you're going to get. So yeah, one pigment was soluble in solvent B, but not in solvent A, because obviously there's one more pigment that came about in solvent B. So there's that one pigment that was soluble in solvent B that wasn't soluble in solvent A. And so I've made 2 points there about both solvent B and solvent A, so that should be enough for 2 marks. So this is the mark scheme. So it says, so for 2 marks, 2 pigments have the same solubility in solvent A but different solubility in solvent B. Or 1 pigment is soluble in solvent B but not in solvent A. So that's what I said. So I'd get both marks for that. So all of these things are kind of just, well, they're saying the same thing but just in slightly different wording. So you can say also that the pigments have different solubilities in both of the solvents. So that's kind of the same thing. And then you can say 1 pigment is only soluble in solvent B. Or you can say 1 pigment is insoluble in solvent A. So yeah, they're all just saying the same thing, really. Then there's this point that you can only get 1 mark for. So you can basically say all those things, but you say solution rather than solvent. So that's not technically right. So you'd obviously only get 1 mark for that. Then the other points then, so they accept dissolves for solubility, because that's technically true, but it's not very scientific. Then correct reference to affinity for solubility. So greater affinity to the solvent indicates more soluble. And then accept correct reference to polarity for solubility. So you can use kind of different terms for solubility. And then also they accept RF value for solubility, but you don't get marks for saying the solvent is stronger, because that doesn't really mean anything scientifically. Right, so that's it then. So that's all I want to say for this video. I hope you found it useful. If you have any questions, obviously please leave them in the comments. And also if you have any requests for videos, then please leave them in the comments. And yeah, I'll see you in the next video.